and hello everyone, welcome to class 12 biology. In this video we are going to discuss about regulation of gene expression. In our previous videos we discussed about transcription and translation, and I told you that those two processes are involved in gene expression. So what is gene expression? Gene expression in simple terms, it means if we have a gene and if that gene produces a gene product, then that gene is said to have been expressed. So we know that gene is the DNA, a small segment of DNA, which codes for the protein. So protein is the gene product. Okay, so in the synthesis of protein, we know that from DNA to protein there are many steps involved. So those steps are transcription, mRNA modification, mRNA transport, and translation. And if you take a typical eukaryotic cell, inside the nucleus of the eukaryotic cell, we can see the DNA over here. The DNA gets transcribed in the first step. After transcription, we have the primary transcript. The primary transcript, it needs to undergo some modifications in which the introns are removed and exons are joined together. And then there is G capping and poly A tailing. And that leads to the formation of a mature mRNA. This mature mRNA, which is formed inside the nucleus, it needs to be transported outside into the cytoplasm, where this mRNA will bind with the ribosomes, the protein factory, and it will synthesize an amino acid chain. And the amino acid chain will undergo certain modifications like folding, and it will lead to the formation of a functional protein. So from gene, that is DNA, till the formation of protein, a functional protein, that is called as gene expression. Okay, so when we talk about how this particular process, the entire gene expression process is regulated, the regulation of gene expression can happen at any of these particulars. So therefore, regulation of gene expression could be exerted at various levels. So those levels are transcriptional level, processing level, transport of mRNA, during transport of mRNA, and during translation as well. So the regulation of gene expression can happen anywhere. During the transcription, when the DNA was copied into RNA, regulation can happen over there. And when the modification of RNA is happening, at that particular level, regulation can happen. And when the mRNA is transported outside into the cytoplasm, at that point also the gene expression can be regulated. And finally, when the RNA is being translated into amino acid chains, if the gene regulation happens over there, then that is the translational level at which the gene expression regulation is said to be happening. Okay. In order to better understand the gene regulation, we will take the example of E. coli bacteria. So E. coli bacteria, inside E. coli bacteria, there is synthesis of a particular enzyme called beta-galactosidase. This enzyme called beta-galactosidase is involved in the hydrolysis of lactose into glucose and galactose. I hope you know that lactose is a disaccharide and glucose and galactose are the monosaccharides which form the lactose. And this particular enzyme, beta-galactosidase, helps in the breakdown of lactose into glucose and galactose are used as energy source by the E. coli bacteria. Now, if we have two bacterial colonies, if we have got two separate bacterial colonies, one which gets lactose in the medium and the another colony, it does not get any lactose in the medium, what happens is, if we observe the one which is getting the lactose in its medium, it produces the enzyme beta-galactosidase. The one which is not getting any lactose in the medium, it does not produce the enzyme. Now, logically speaking, there is no need of the enzyme over here because there is no lactose. If the substrate is not there, what is the use of the enzyme? And somehow, if the bacteria produces that enzyme, it's a wasteful process, right? So the bacteria knows somehow that there is no lactose, therefore there is no need to produce the enzyme, right? So over here we can clearly see that the metabolic, physiological, or the environmental conditions of the cell regulates the gene expression within the cell, right? So a particular E. coli bacteria under the influence of the environmental conditions, it can regulate its gene expression. So the gene which is responsible for the production of beta galactosidase, it somehow switches off when there is no lactose present, and when there is lactose present, that gene switches on and starts producing beta galactosidase. Right. So over here we can see that the metabolic, physiological or environmental conditions regulates the gene expression. And in this particular case, in E. coli bacteria, the production of beta galactosidase, the gene regulation, happens at transcriptional level. So how do we know that the gene regulation in this particular case happens at transcriptional level? We know that because of the work of these two scientists, Jacob and Monod. So these two scientists, they came up with a particular explanation called the lac-operon regulation. So I will explain what is lac-operon later on. First, let us try to understand what they were doing, right? What these two scientists were doing. Jacob and Monod, they studied lactose metabolism system within the bacteria. How lactose was metabolized within the E. coli cell. They were studying that, right? And they were trying to figure out how the bacterial cell knows which enzyme to synthesize. The gene regulation of the lac-operon. So don't worry about this. Lac-operon, I will explain later on. The gene regulation of the lac-operon was the first genetic regulatory mechanism to be understood clearly, right? So these two scientists, they explained how lactose metabolism happens within the bacterial cells. They try to figure out how the cell knows which enzyme to synthesize. That means they are trying to figure out how the genes which are involved in the lactose metabolism is regulated. So the gene regulation of the lac-operon was the first genetic regulatory mechanism to be understood clearly thanks to these two scientists. Therefore, these two scientists were awarded the Nobel Prize in Physiology in 1965. Now let us try to understand what is lac-operon. What is the thing that they try to explain? An operon, first of all, the definition of an operon is a functioning unit of DNA containing a cluster of genes under the control of a single promoter. Right. Over here you can see an operon. So this is an operon, just like the transcriptional unit. Here also we have a DNA segment, a functioning unit of DNA, right, containing a cluster of genes. So we have a promoter gene, operator gene, structural genes, and then we have terminator gene also over here. It's not shown here, but there's a terminator gene as well. So we have got all these genes in a cluster, and all these genes are under the control of a single promoter, this green one. Right, all these genes, operator and structural genes and everything, they are under the control of a single promoter. So therefore, since they are under the control of a single promoter, the genes contained in the operon, in this entire operon, are either expressed together or they are not expressed at all. Okay. And in our case, we have to study the lac-operon. So we will take a look at the representation of lac-operon. So here we have a lac-operon. From here to here, it's a lac-operon, but we have to write this extra gene over here, the I gene, because it is involved in the control of the expression of the operon. So here the P stands for promoter. So both these P stands for promoter. And here the I stands for inhibitor. O stands for operator. And Z, Y, and A, they are all structural genes. And these structural genes, they represent or they code different enzymes. We know that in prokaryotes, the structural genes are polycistronic. That means they code for more than one protein, right? So here, these structural genes, they code for 3 different proteins or 3 different enzymes. So the Z gene codes for beta galactosidase, the enzyme which is responsible for the breakdown of lactose into glucose and galactose. And then we have this Y gene which codes for permease enzyme. The permease enzyme increases the permeability of the bacterial cell wall and cell membrane towards beta galactosides. And we have the gene A which codes for transacetylase. And now this enzyme transacetylase, it does not have there is no known function of transacetylase in lactose metabolism, but since it is attached with the lac-operon, therefore we have to know the name of enzyme transacetylase, which is coded by the gene A. So this is lac-operon. Okay. So the lac operon simply means the operon which is involved in the lactose metabolism is called as the lac operon. Let us now try to understand how this lac operon is regulated. In order to understand the regulation process, you need to understand this particular diagram thoroughly. You should study this particular diagram given in your textbook thoroughly, and you should be able to explain this particular diagram if it is given in your textbook, your examination. There are two scenarios given over here. One scenario, it is in the absence of inducer. In the second scenario, it is in the presence of inducer. So the lac operon in the absence of inducer is the first scenario, and the lac operon, same lac operon, in the presence of inducer is the second scenario. So I will explain both the scenario one by one. So in the first scenario, in the absence of inducer, so now here the inducer is the lactose. Okay, so in the absence of lactose, when the bacteria is not getting any lactose, in the absence of lactose, the operon, what happens is the I gene or the inhibitor gene, it expresses, the RNA polymerase binds over here and it moves and it synthesizes a repressor mRNA by copying down the I gene or the inhibitor gene. By looking at the inhibitor gene, it produces a repressor mRNA. The repressor mRNA will be translated into a repressor protein, and in the absence of lactose, this repressor protein goes and binds to the operator of the lac operon. So I will show it again. The repressor protein goes and binds to the operator region of the lac operon. So what it does, when the repressor protein goes and binds with the operator, the RNA polymerase which goes and binds to the promoter of the lac operon, it cannot move in this particular direction because its way is blocked by the repressor protein. So the presence of repressor protein over the operator region prevents the accessibility of these region towards the RNA polymerase. So therefore, the gene Z, Y, and A are not expressed. They are not transcribed. So in the absence of lactose, what happens? Repressor binds to the operator and prevents the RNA polymerase from transcribing the operon. The entire operon is not transcribed. This particular situation is said to be switched off, right? This particular state is said to be in switched off state. This lac operon is said to be in switched off state because there is no production of any enzymes over. Now the second scenario, in the presence of the inducer. So I told you that inducer is the lactose. So we have over here the inducer. In the presence of inducer, when the bacteria is getting lactose, what happens? The same thing, right? The production of repressor is there. Now over here, the inducer will go and bind with the repressor protein. The lactose which is present over here, it goes and binds to the repressor protein and it makes the repressor protein inactive. Right? The binding of the inducer to the repressor protein makes the repressor inactive. It means that the repressor protein can no longer go and bind to the operator region. And now since the repressor cannot go and bind to the operator region, the RNA polymerase can go and bind with the promoter and then it can transcribe the entire structural gene. And when the structural gene is transcribed, it produces an mRNA, the lac mRNA. And from this lac mRNA, we get beta-galactosidase, permease, and transacetylase through translation process. I told you that gene Z codes for beta-galactosidase, gene Y codes for permease, and gene A codes for transacetylase. So 3 enzymes are produced. These polycistronic structural genes, they lead to the formation of 3 different proteins or 3 different enzymes over here. This particular state of operon is said to be in switched on state. So the lac operon is in switched on state when there is presence of lactose. So that is what the diagram is trying to explain. I hope you are clear about this one. In the lac operon, what happens? In the absence of inducer, the operon remains switched off. Why it remains switched off? Because the repressor protein, the accessory repressor protein, which is produced by the inhibitor gene, goes and binds to the operator region of the lac operon and it prevents the RNA polymerase from transcribing the structural genes. Therefore, there is no production of enzymes. In the second scenario, in the presence of inducer, when the lactose is present, what happens? The inducer goes and binds with the repressor protein, makes the repressor protein inactive, and therefore the repressor protein is unable to go and bind with the operator, thereby making the entire region free for the RNA polymerase to transcribe. LEC mRNA is produced, and from this LEC mRNA, these three enzymes are produced. And this one is said to be in switched on. The LEC operon is in switched on. So that is about the LEC operon. And I hope by knowing how LEC operon is regulated, you can see that this particular operon is regulated at the transcriptional level. So this regulation is happening at the first level, that is the transcriptional level. And there are many other examples like tryptophan operon, right? And many other examples are also given: valine operon, right? Histone operon, tryptophan operon, right? So what I want you to do is, for your competitive examination's sake, I want you to look through the internet and try to study about the tryptophan operon and try to compare the differences between the LEC operon that we have studied now and tryptophan operon. What's the difference between the two? So that should cover the topic about gene regulation. Okay. So next video we are going to discuss about Human Genome Project. So the same thing, I want you to read the entire portion on Human Genome Project given within your textbook, and then you can watch the next video. Thank you.